All right. How's it going? Today is the last class of Bioengineering 101. Um, because it's the last class and we've covered most of the material, we're just going to hang out, drink a beer, answer your questions. If you have any troubleshooting questions or problems, let me know. If you have any science questions, just don't ask me anything about living forever or some bullshit like that. Um, you know, uh, make it some good questions, and I'm down to listen. I'm down to answer. Um, we have a lot in store for future ODIN classes that we are pretty excited about. Um, for all the people who have taken this class and learned how to use a pipette and learned how to do genetic engineering, um, we're looking at starting a human cell culture and gene and editing class. Yeah, we're working on it right now. Hopefully we'll be out in the next couple months, which is pretty exciting. Hey Kent, what's up? I'll be right back. Somebody rang the doorbell. Okay, I'm back. Sorry about that. All right, what we got? Lots of questions. Sorry to run away. I had somebody who came to the Odin and wanted to buy some stuff with cash. Doesn't happen often, but sometimes happens, and you know, pretty good. Okay, so here we go. The guy who's working on glowing puppies. Can you explain how it's possible that puppies can produce the glowing protein through their bodies? So, if you're working on glowing puppies, what he's trying to do is he's trying to engineer the puppies at an embryo stage. So, he's trying to put DNA into the embryo. If it's in the embryo, then theoretically it should spread to every cell. If it works. The problem is they've never really made any sort of animal be able to glow. So who knows if it'll work? Maybe. They use viral vectors instead of plasmids. When will the next Bio 102 be available? Kent, we're probably going to start the classes up again in August for Bio 102. We might revamp it a little bit. Right now, people are having some difficulty with it. I think we made the class too complicated, and we need to tone it down a little bit. But it's been going pretty good. But yeah, I think we need to tone it down a little bit, make it a little less complicated than what it is. Hey, Dixon, in Cary, North Carolina. I actually lived in Boone, North Carolina for a while, and Chapel Hill. Any about CRISPR for HIV? Using CRISPR on things like HIV or herpes is going to be difficult. Not because CRISPR doesn't work, but you have to understand that, like, in order to really get rid of HIV or get rid of herpes simplex virus or something like that, you have to um, get every cell, right? So there's all these cells, these nerve cells, and you have to engineer and edit all of them. If you don't, then the virus is still going to be around. Um, I think the most likely advancements for things like HIV and herpes won't be using CRISPR, maybe using a vaccine or gene therapy or something like that. Kent, yeah, Kent says, in regards to glowing mammals, would the body temperature denature the glowing protein? Wouldn't there be better luck trying it on things like reptiles? Yeah, so there are a couple different types of luciferase proteins that do make things bioluminesce. In our class, we used one of them, and that was a bacterial luciferase, and it's one of the best ones because it actually part of that genes that's involved in it, they produce their own substrate. So usually when they use luciferase in mammals or animals, what they have to do is they have to add this compound that's called luciferin. Now, luciferin is the chemical that luciferase uses to actually make the light or the glow. So they have to add it exogenous from outside. The genes can't produce it. So that's a huge problem. Um, it might be interesting to try other things like reptiles. I don't know if anybody's tried it. You should search the scientific literature and see. Um, well, I think I sent out a questionnaire about people wanting to work in the industry. So what we've been doing is we've been having a jobs program. Victoria Santos, who said hi earlier, she's part of that. What we're trying to do is we're trying to help people get jobs in biotechnology by helping them update their skills, um, update their resumes and interviewing skills and all these different things to help them get jobs. Um, so yeah, we're gonna see how that works out and see if we can expand it and help out more people who have some biotech skills either through our classes or through schooling and want to get a job. Raul, CRISPR using transposons is not that good because the accuracy isn't that great. We can already insert genes like that into cells and things in many different ways. It's not as amazing as you imagine. Whenever there's a news article about CRISPR, be extremely skeptical, right? People are hoping that using the hype of the word CRISPR will get clicks, and it does. But often the research is never really used, right? So I mean, CRISPR, normal CRISPR with Cas9, uh, it's not even been used in the clinic yet in humans, right? And so like the chances that any of this other stuff is ever gonna do any great thing is very minimal. Can you contact me about the jobs program? Yes, you can. I don't know what will come of it right now, so but you can send me an email. My email is case at the-odin.com, and contact me if you want, and we'll see what I can do to help. Yeah. Originally, we were looking just in the Bay Area. That's where we're based. The Odin is in Oakland, California. And originally, we were looking just in the Bay Area so we could leverage our network here to try to help people get jobs. Um, but I think it's going to be, uh, I think we might expand because I think it's going to be less leveraging our network and more uh, just giving people job skills. Alexis, in the case of the plasmid PJE202 for bioluminescence, the effect is attained only by expression of those genes. Yeah, so it is attained only by expression of those genes, but you have to keep the temperature below a certain level because the protein or the chemical reaction doesn't work above 30 degrees Celsius, right? So it does need certain environmental conditions. The chemical conditions aren't so important as long as it's, inside the cell and in proper conditions. But yeah, that's all that's needed basically is a temperature. And you know, the bacteria start to lose their bioluminescence after a certain amount of time. Um, But if you add more media, add more antibiotic, you could keep the bioluminescence going. You just have to keep, refreshing the media and right because they need food, they need different things to keep going. All right. What else? What else questions you got? yeah, one of the things we really want to do is we want, to help people get more out of these classes than just learning genetic engineering. We want to help them get something for themselves, like a job or, um, you know, get promotion or something. So it's something that we're working a lot on. The next course. We got a couple courses we are planning. It's just, getting people to work on them. So we have 3 classes in the works right now. One class is a class on how to use PCR to do amplified genes for DNA fingerprinting of different organisms. So basically, PCR is a technique, polymerase chain reaction, uh, that it allows people to amplify DNA, and when you amplify DNA, then you could do whatever you want with it. It's a lot easier. Like, you can't just sequence the DNA from your cells. What you do is you extract the DNA from your cells and you amplify it. Instead of sequencing all the DNA, what people often do is they take a fingerprint, right? So when people use DNA sequencing um, for criminal stuff, it's not actually, they don't sequence the genome of the person or the cells. What they do is they do this fingerprint analysis of 13 different genes, right? Well, you could do that with all things. You could do it to discover and identify new plants, um, new mushrooms, new animals. So we want to run a class that teaches people how to sequence and identify and contribute to the public databases for DNA sequencing identification of organisms. Uh, so hopefully we'll have that class out in the next few months. The next class is a mushroom cultivation class. So it's teaching people how to work with mushrooms, how to take spore prints, how to grow mushrooms, how to, identify mushrooms, how to do all stuff cool with mushrooms. Um, and hopefully that one. And then the other class we're working on is human mammalian cell culture. So when you're culturing not bacteria or yeast, but human cells or mammalian cells, it's a bit different than other types of cell culture. And it's more complicated and there's more things going into it. But as you can imagine, there's so much more you can do with human cells than you can with bacteria or yeast just because it's human cells. So right now we are in the final testing stages of a class that teaches people how to culture human cells and genetically modify them. So that'll be a really cool class. Um, it's just, getting all this stuff done and out there. Um, yeah, expanding all the classes so people have a lot more to choose from and work on. When do we expect to start that class? Oof, I'm gonna try to aim for August and start registration in July, but it depends on a lot, so we'll see. But that's the goal, to start the class in August and start registration in July. Um, Yeah. Alexis, can we use AAV vector cultured in human cell line and mice? Um, yeah, AAV vector should infect any sort of mammalian cell. Um, all right. So what else is going on here? Oh yeah, if y'all don't know, we have a conference that we run called Biohack the Planet. And Biohack the Planet is August 31st and September 1st. And it's going to be in Las Vegas this year, and it's going to be awesome. It's going to be a lot of biohackers and scientists and media people who are going to be there and, you know, talk about biohacking, just have fun, party hard, enjoy everything. And it's a great opportunity to meet people in the community, um, to get involved in the community, to, you know, learn what's going on. So I would look into it, even if, you're busy around that time. It's a Labor Day weekend, I think. Labor Day, right? Is that August 31st, September 1st? So Saturday, Sunday, and then I think Monday most people have off for Labor Day. Uh, can hang out in Vegas. There's lots of cool stuff to do. And you know, just come out. Tickets are $150, and I think a hotel room is like $100 in the hotel that we're doing the conference at. So, you know, if you if you want, just split the hotel room with somebody and, make it really cheap. Tickets are really cheap to Vegas, almost everywhere in the US. Probably almost everywhere in the world, tickets are cheap to Vegas. So get to Vegas, join us. You can go to the conference is called Biohack the Planet. So you can go to like Biohack the Planet. We usually live stream the talks, um, but we're going to have poster sessions and other things that won't be live streamed. So you will miss a lot if you don't go. But yeah, we should hopefully live stream the talk. Biohacktheplanet.com. Check it out. Any any thought on how engineers can overcome the inability to affect all cells in a gene therapy? Yeah, I mean, you can affect all cells theoretically. It's just you have to get a virus to all cells, right? And that's the tough one, or get the DNA to all cells. And the human body is huge, so trying to get it would be a feat to just get something, to get anything to every cell in the human body. But the thing is you don't need to modify every cell, right? No matter what you're doing, you generally don't need to modify every cell. If you're talking about anti-aging bullshit, take a step back. We're not talking about that stuff. I don't think I don't believe in it. Um, but you don't need to target every cell. Usually, all you need to do is target enough cells to have an effect, right? It's like enough cells to have the effect. Once you have the effect, then it doesn't matter. So that's generally what you're trying to do. Just target enough cells to have an effect. All right. What else you got? Ooh. No, I mean, when you genetically modify cells, the body doesn't see them as abnormal at all, Raul. Um, body doesn't care if you genetically modify cells. Body has no response generally. Um, yeah, no problem. I mean, you have to understand they're your own cells. Your body usually doesn't respond to your own cells, right? And that's why cancer works. That's why cancer kills us, is because cancer is our own cells going out of control. And so our immune system has a hard time responding to that. I mean, that's what causes autoimmune diseases, also, right? If you have an overactive immune system, and then your body has autoimmune reactions, right? So that's usually what happens with an overactive immune system. Your body has autoimmune reactions. Not gene therapy to attack cancer cells, Alexis, but you have to you have to modulate the immune system. So there are things like immunotherapy and checkpoint inhibitors. So what these things basically do is immune checkpoint inhibitors, what they lower your immune system's threshold for what they should attack, right? So you can imagine if your body doesn't normally attack uh, normal cells or like cancer cells, what you can do is you could lower that threshold a little bit, and then your immune system starts to recognize cancer cells because they're different enough. The problem is, again, that can cause autoimmune reactions and things like that. But disease, drugs like Keytruda is supposed to be one of the next big blockbuster drugs. It's been out for a little bit, um, and it works really well in some cancers. Yeah, by checkpointing the immune system. But immunotherapy is the general field for attacking cancer in regards to like immune reactions. So if you are interested in stuff like that, look up immunotherapy, and you should be able to find lots of info. Now, Raul, I mean, you could change all your cells in your body, and that doesn't equal cancer. Cancer is when your cells, certain cells in your body, replicate uncontrollably, right? So basically, what chemotherapy is a drug, and what it does is it stops all the cells in your body from replicating, right? So when you have chemotherapy, that's why your hair falls out, because you have replicating cells that are causing your hair to grow and things like that. That's why your intestine, stomach, gastrointestinal tract, that's why that suffers a lot of issues because those mucous membranes, you have replicating cells in there, right? So the rest of your body, generally the cells aren't replicating all the time, so it doesn't hurt them as much. Whereas cancer cells are trying to constantly replicate. So if you can stop them from replicating, it kills off the cancer cells, and you know, it stops them from spreading. But That's what differentiates cancer from just a genetic change in the cell, right? Hopefully one day we'll have a class on how to make your own gene therapy. But I don't want to be, thrown in jail right now by the government. So we'll stick away from that for a little bit. Come on, people. I need more questions. I'm finishing up my beer. Not talking that much. I'm going to finish my beer early. Do I recommend going to school? It depends what you want to do. I think I made. Did I make a YouTube video about that? Let me see if I can find it. I think I made a YouTube video about that. Should you go to school? And it really just depends on uh, what you want to do with your life. Here's a video I made.
What's up, everybody?
You can check it out. School never hurts, but it does take time and effort and money. So it really depends what your goals are. If your goals are to get a job in biotech and you don't have any college, you're probably going to have a hard time. So I'd suggest, going back to school for an associate's degree or something like that, if you can, a bachelor's degree even. Uh, but if you're just trying to do research and learn genetic engineering on your own, you know, the time is probably better spent just, learning things on your own. It's probably, I mean, you have to understand in school, you're not going to get to do much interesting stuff. What you're going to get to do mostly is just, you're just going to have to take classes and tests and stuff like that. Is it possible to make something that would make food discolor when expired? Like a yogurt that would turn blue when it began to mold or something like that? I think the problem with that question is that what does it mean when food expires, right? I think that means a different thing for every food. And so that's Victoria, it depends really what you want to do. Um, if you want to, you know, the thing is that nowadays a lot of times what you can do is experience will function in place of a PhD or something. So I know a lot of people, or just some people who have gotten PhD level positions just because they've been working in the biotech industry for the same number of years it takes to get a PhD, right? So a lot of places, if you have 5, 6, 7 years experience, they will take that in place of a PhD. Um, if your goal is to do science and make money, you probably don't need a PhD. If your goal is to get a title, try to work in academia or something like that, then you're going to need a PhD. Uh, I, you know, if I had to do my PhD all over again, I don't know if I would do it. Uh, I mean, it wasn't the most terrible time in my life, and most PhDs are paid for, right? So most PhDs, most universities in the U.S., they pay you a stipend, and you don't have to pay tuition. If they don't pay you a stipend and they don't waive your tuition, then don't go to that school. But, you know, you get paid to do a PhD. It's not much. Most schools will probably pay you between 25 and $35,000 a year. Uh, but it's, generally enough. But then understand you're going to have to do it for 5 or 6 years. Um, and when you get out, you're not going to necessarily get paid much more, right? So leaving a PhD, doing a postdoc, your starting salary for a postdoc is around $40,000 to $50,000. Your starting salary at a company with just a PhD and no postdoc is probably around 70 or $80,000. If you do a postdoc and you have a special skill, you might be able to push it up to about $100,000. But then you have to understand if you were just to work in your field for 7, 8, 9 years, you probably can make the same amount of money, right? So it just really depends what you're going for. Yeah. So, Kent, like we talk about, and if you look at our syllabus, uh, you could get by any paywall if you use sci-hub.tw. Um, that should uh, that should get you by most paywalls. I mean, honestly, learning there's not many places you can learn about bioengineering right now. Uh, this is one of the only places. Yeah, I think Federico was right. It's probably more of a chemical thing. I think there was people who were trying to develop like a sticker that you could like put on fruit or vegetables that responds to the hormones or chemicals released when they start to deteriorate and change the color of the sticker or something like that. Yeah, I don't know. It's an interesting question. Do some research into it and see if you could come up with anything. DIY. Make a proof of concept, and then, maybe you could raise some venture capital funding or something like that. Yeah, I mean, Lex is on hiring abroad and stuff like that. It's not necessarily a financial thing. Um, I don't think that's it. It's more of just people in the biotech industry aren't comfortable hiring people without experience, right? biotech has always been this thing that only existed in the academic world, and we're trying to change that, right? We're really trying to change that. And so helping people get jobs without experience, but with hands-on skills, is something that's really important. Once that starts happening, I think a lot will change. Um, because let's be honest, it's cheaper to hire somebody with a bachelor's degree who has the skills of a PhD, right? It is. That job is always going to be paid less to the person with the bachelor's degree. So you could help a lot of companies, a lot of places by providing skilled labor that's not doesn't have fancy degrees. Um, it would help a lot of companies, a lot of places. Um, but it's convincing people to trust these people without degrees, right? Because everybody thinks, oh, like you need a PhD to do X or Y or Z when you really don't, you know? You really don't need a PhD. People just need hands-on experience. And usually the only way to get hands-on experience is in an academic setting. We're trying to change that, right? Everybody in this class is trying to change that. It's like you don't need to be an academic to get experience anymore. You could get it in your home. You could work on this stuff in your home. And I think that's going to change a lot. It's going to change a lot. A lot is going to change because of that. But it's going to take time, right? These things don't happen overnight. It takes time. So, who knows? Hopefully in the next year or two we can start seeing people uh, who don't have degrees getting jobs just based off their experience and knowledge. That would be awesome. Yeah. Yeah, we'll probably provide people with a certificate for these classes that you're taking right now. So expect a certificate through email. We might have some kind of test you have to take or something like that to get the certificate. But you know, uh, we we plan on providing that to people. Even though it probably doesn't mean much right now, we hope in the future it does mean a lot more. We're just starting out. This is only our second class, our second time teaching this class ever, right? So it's gonna take a little bit for it to start carrying some weight, but we hope it does. We really hope it does. Yeah, we want to set it up so that people can put it on their resume or CV and let people know that they took these classes, right? Yeah, we have to build up a name. So that means y'all have to go out there and do some pretty cool stuff, right? Because once everybody starts doing cool experiments and people know that got training at the ODIN school, then we won't have to. It'll be all promoted for us. So that's what everybody's job is. Go do some cool things. All right, people finishing up. Do not have any more questions? Yeah, come to Biohack the Planet. It would be a great time. Great speakers. You get to meet so many cool. I mean, you get to meet me. Come on. I'm joking. I'll probably be the least coolest person there. Probably be so many other way cooler people. HB Wilder, for the fungi class, what does the price supplies approximately come to? I don't know yet. Um, that class is still in the early stages of planning. It'll probably be cheaper than these other classes because there's less supplies that are required. Um, but we'll see. Yeah, it'll probably be cheaper. We're currently, ooh, any thoughts on prebiotic chemistry and chemical evolution? How the first cells evolve? A lot of thoughts. Um, They'll probably take too long to talk about. Says he's currently a machine learning engineer at a hedge fund. Is there a high demand for my type of skill in bioengineering? Uh, um, in genetics, maybe a little bit. There's not really a high demand for uh, it's not really a high demand for machine learning in bioengineering right now. Victoria, do we need volunteers for Biohack the Planet? Yes, we need volunteers. I think on the website, if you go to the website, there's probably an email that you can email about volunteers. Um, we can give you free tickets, but we can't pay for your flight and stay if you volunteer. So you'll have to cover that on your own. But yeah, check it out. Glad you enjoyed the class. Ben, you know, thinking of going back to school for bioengineering. It really depends what you want to do, you know, Ben. We talked about this. If you scroll up in the chat, I posted a YouTube video that I made about going to school and whether it's good or not. You know, if I were you, I would just start learning about bioengineering. If you got a job with machine learning without going to school, you get probably going to get a job in bioengineering the same way, right? Learning enough about it to show that you're functionally competent. That's what you got to do. So I would say you probably have a better chance of doing that without going to school than going to school. Um, Unless you have like all your prerequisites out of the way and you know a school that will allow you to finish up in like 1 or 2 years taking bioengineering courses. Um, I probably wouldn't do it. You know, it's probably not going to put you in the position that you want to be in. All right. Anything else before I finish this beer and take off? I've almost been here for an hour. Last questions. Hit me. Let me have it. I'll do my best to answer. Anything. Anything. Well, as long as it doesn't take like an hour to explain. Or as long as it's not about something crazy like longevity or some bullshit. Everybody wants to live forever. I don't get it. Would it be possible to alter your eye color? Possibly, but you have to inject some shit in your eyeball. Probably not something you're interested in. And if you are interested in injecting shit in your eyeball, you know, just wear colored contacts or something, right? They're they're inexpensive. It's a lot easier. No risk of permanent damage. As far as eukaryotes, protists, you can't put PGA202 in yeast because we learned in the class that there's different DNA elements for different organisms, right? So something that works in bacteria won't work in yeast, won't work in humans. So no, you cannot. As far as eukaryotes are concerned, what are the easiest to transfect? easiest to transfect are you know, eukaryotes aren't difficult to transfect, right? They they're all fairly easy. I mean, yeast is a eukaryote, right? Uh, plants are plants and fungi are more difficult because they have a cell wall, but most other things are. It's not difficult. It's just different, right? It's different. So it's finding out what works. Bacteria are really easy compared to all their cells. So it's just different. Um, yeah. And hopefully, like I said, we'll have that class, mammalian cell culture, and transfection, and genetic engineering, which will be really cool. Anyway, I'm going to take off. Thank you all for being here in this class. If you have any questions, we'll still be around on Google Classroom. Ask your questions on the Google Classroom. Otherwise, find me on social media. You know, send me a friend request on Facebook or something like that. I'm around sometimes to chat. Hit me up. If I don't have time to chat and don't respond to your request, don't hate me. Uh, I'm really busy, but I'll do my best. Anyways, look for the other classes. We should have open registration pretty soon. So if you're looking for next class to do, yeah, check it out. We should be opening registration for more classes soon. Otherwise, I will talk to you all later.